as the second kind of lecturer of this academic year. Meister was born in Germany, grew up in Italy, and then studied physics back in Germany, in Munich. You must be Jewish with this trajectory. And finally finished his PhD in physics in 1987 in Caltech, where he studied bacterial locomotion under Professor Howard Berry. He then began his career in neuroscience with a postdoc fellowship at Stanford in James Baylor's laboratory, where he studied issues of development of the retinal circuitry, and there he also developed the famous multi-electrode array system for recording from the output of the retina, and I'm sure we'll hear more about it today. In 1991, he moved to Harvard as an assistant professor of biology, and finally became a full professor at Harvard in 1998. Markus has many honors and international recognitions and serves as on the advisory board of Princeton University Department of Molecular Biology, Cold Spring Harbor, Max Planck Institute for Neurobiology, and many others. He publishes almost exclusively and most prestigious and highly visible journals such as Science, Nature, Neuroscience, Neurons, Biology, etc. He actually started his career in neuroscience with a short but very influential letter to science in 1990. The title is Postdoc Tuition at Stanford, signed by Markus Meister and colleagues. So that's the beginning. But of course, he's recognized internationally for his other contributions. He is world leader in sensory processing in the early stages of the visual and olfactory system. Among his major contributions are the development of the multi-electrode array, which became the standard tool for studying of the retina. His discovery of rich adaptation phenomena of the retinal circuitry, his finding that the neural codes in the retina work not simply through neurons which are activated or inactivated, but through complex firing patterns such as synchrony and latency. He is innovative and daring and continues to develop and use new techniques in his research, such as wireless telemetry, optical imaging, and more recently transgenic markers together with George Sainz and colleagues, which led them to the discovery of a new type of retinal ganglion cells which are selective for direction of motion. His view of the brain or the neural circuitry is unconventional. According to Meister, the neural circuitry is not simply hardwired by the structure, but it's dynamic, it's context dependent, and always function oriented. Most importantly for us at the ICNC, Markus combines very uniquely superb electrophysiological techniques with strong insight and deep understanding of the underlying computational issues. Well, he was a physicist by training, so it's not surprising to us. So Markus established himself as one of the world leaders of the fledgling field of computational neuroscience, and serves as a role model for the rest of the neuroscience community for a successful implementation of theoretical and computational ideas into neuroscience. His conviction that understanding the brain requires out-of-the-box approaches and convergence of skills from different disciplines led Professor Meister to initiate about 8 years ago the establishment of Interdisciplinary Center for Brain Sciences at Harvard, and to become one of its major leaders to this day. So it's a pleasure to welcome Markus, a close friend, also young and original influential scientist, and as according to our tradition, give you a diploma or a certificate. The Heller Lecture Series in Computational Neuroscience Awards this certificate of recognition and appreciation to Professor Markus Meister for his contribution to academic inquiry and exchange at the Interdisciplinary Center for Neurocomputation at Mount Summer campus in Los Angeles, Israel, signed by me and by Professor Melkin, head of the PhD program. And I would like to, before stepping down, I would like to mention and remind all of you that today's lecture will be, I guess, primarily about retinal processing, about vision, and tomorrow at the ICNC at 5 o'clock, he will continue his lectures with a seminar on function.
Thank you so much, Haim. Thank you for this overly flattering introduction, but more importantly for the invitation and the great honor to be able to speak in this renowned lecture series. It's also given my wife and myself an opportunity to come and visit this country, and we've already enjoyed it tremendously. We've been here for about 10 days, 2 weeks now, and visited wonderful places and met wonderful people. At the same time, I must say that after 10 long days of vacation, it was refreshing to talk about science again this morning. And there are just wonderful things going on here at the center. I've really had a great time with your colleagues here. Like many of you here, I've been working and thinking about the function of large circuits of neurons. And in some sense, I think that's where the deep mysteries of brain function lie. What happens when many neurons get together, hundreds of thousands of neurons, and form many synapses? What happens in these networks that is somehow different from what happens in 100,000 liver cells? Yeah? There's something about the connectivity and collective function of circuits of brain cells that is different from the liver and the kidney. In trying to figure out how to work, it I think helps to think a little bit ahead, like maybe one generation ahead, and ask yourself, what would you like to know eventually? What would we want an undergraduate textbook of the future to say about this kind of central mystery of brain function? And I think one would like to understand neural circuits in fundamentally 3 different ways. They're complementary, of course. At one point, if you pick your favorite neural circuit, maybe the hippocampus or a cubic millimeter of cortex or some nucleus elsewhere, you would like to know in some fundamental way, what does the circuit do? What are the inputs to that circuit? What are the outputs from that circuit? And what are the transformations in between that turn the inputs and the outputs? Secondly, you'd like to understand how these functions are implemented by the neurons and synapses. So what are the actual mechanics of this processing? And that relates the circuit function to the kind of lower level of molecules, cells, and synapses. And thirdly, you'd like to understand how this little circuit plugs into the overall brain function. You'd like to understand why nature built it this way and not any of 100 other ways, and how it plugs into the brain as a whole, and how it helps the animal survive and implement its behavior. So that relates the local circuit to the bigger picture of the brain, the mind, and behavior. So we would like to, in some sense, have, for any given brain circuit, we'd like to have some insight on these 3 topics, I think. We've been trying to develop this kind of understanding working on the retina. The retina, you remember, is the neural circuit that is in the back of your eye. This is a section through the eye. The rest of the eye is a camera, essentially. Up here is the aperture and the lens, and together they form a picture of the outside world on the back of the eye, and lining the back of the eye is the retina. And the retina serves the role of the film in the camera, but it does much more than the film in the camera. It really begins to process this image in a lot of interesting ways, and sends the results of this processing on to the brain through the optic nerve. So if you take a little cut out of the retina and look carefully, you find that it is a layered circuit. It has this layered structure. At the outermost layer are the photoreceptor cells. These are the neurons that actually absorb the light and turn it into a neural signal that can be processed further. They talk to a set of neurons called bipolar cells, and the bipolar cells in turn make connections to a population of neurons called ganglion cells. These are the output cells, so they send their fibers to the brain, and the action potentials, the little electrical spikes on those fibers, are all the signal that your brain receives about the visual world. So everything you experience in vision derives from little electrical pulses that travel along these fibers to the brain. Now, in addition to this vertical pathway straight through, there's a lot of horizontal interaction at this layer of synapses by the horizontal cells. They connect the photoreceptors and bipolar cells. And at this level of synapses by amacrine cells, they make horizontal connections down here. And as a result of this intricate circuitry, the retina really performs, as I mentioned already, a good amount of processing, and we'll find out more today what the nature of that is. From a technical standpoint, the retina is really a great playground. It's a great circuit to use as an object of investigation because it offers so many advantages. So one advantage is that we have wonderful control over the input to the circuit. You can control the signals in about 100,000 photoreceptors just by shining a pattern of light on the retina in your experiment. Yeah? And so we do that by taking the retina out of the eye and projecting onto it an image from a computer monitor that drives each of these photoreceptors with a different level of light. Yeah? So this gives you access to the input of this neural circuit. At the output, these retinal ganglion cells are the output neurons. They're all arranged in a convenient layer here. And what we can do is put the retina into a dish that has implanted in the bottom many small metal sensors. And each of these sensors is an electrode. And when one of these ganglion cells fires an action potential, the electrodes can pick up these signals and record them, and we can then analyze them. So these signals would be the ones that normally flow to the brain, except we intercept them now and try to understand how the retina signals the image that was presented to the photoreceptors. Now, we also have a number of tools to influence the circuitry within the retina. We can use drugs, pharmacology, to affect the way the neurons can communicate to each other. We can also use additional electrodes, like traditional sharp glass electrodes, to puncture individual neurons and record their signals or manipulate them. And increasingly, we have also genetic access to the circuitry. By working with transgenic mice, we can modify the circuit in the retina. We can remove certain neurons. We can change their properties. We can silence them, activate them. And that gives a lot of access. So, for example, by eliminating one amacrine cell, gives us a lot of access for manipulation that in turn serves to test our ideas of how the circuit works. So, let me tell you a little more about what's known about the structure of the retina. This circuit, even though it's considered a simple neural circuit, it contains about 50 different types of cells, 50 different types of neurons. Yeah? At the output layer, there are about 15 types of retinal ganglion cell, and they differ a lot in the size of their dendritic fields, so whether they collect input from a large region of space or a small region of space. They differ a lot in their temporal properties. Altogether, about 15 different types. Now, each type of ganglion cell forms a complete tiling of the visual field, meaning each type actually conveys a complete image to the brain. And so the retina really produces about 15 types of neural image that get conveyed to the brain in parallel. Sometimes they go to different areas of the brain. Sometimes they merge in the same area. So in some sense, the long-term goal of retinal neuroscience is to understand what these 15 kinds of computations are. What are the 15 type of image operations that the retina performs on the raw visual input? Looking higher up, we have 3 or 4 types of photoreceptors, about 10 types of bipolar cells, about 30 types of amacrine cells. So there's a great diversity of components in this circuit, probably similar to the number of cell types you would find in a local area of the cortex. And furthermore, these cells are interconnected in very specific ways. So for example, this type of bipolar cell may connect only to this kind of amacrine cell and this kind of retinal ganglion cell. So the circuit connectivity is highly specific, and this is just an image to illustrate that. This is a section through the retina, and we have here, between here and here, the so-called inner plexiform layer. That's where synapses are formed between bipolar cells, amacrine cells, and ganglion cells. And these colorful stains are antibody labels that selectively label certain types of ganglion cell or amacrine cell. And they show you that these connections reside exclusively in these thin laminae, these thin layers of this inner plexiform region. So they are restricted, very highly restricted, to just a subset of the possible synapses. So in summary, 50 kinds of components, and they're interconnected in very highly specific ways. So beautiful complexity to the structure of the circuit. So what about the function of the circuit? What do the textbooks say about the function of the circuit? And I'm in the process of revising one of those textbooks, so I can tell you what the last edition said. It said that the retina performs lateral inhibition. The individual ganglion cell is excited by light in the local patch of the image and inhibited by light in the surrounding region. So in some sense, it computes the contrast between the local intensity and the surrounding intensity. And that operation should serve to highlight edges in the image and highlight contours of objects. So this is the standard, the conventional wisdom that the retina's function is to perform this center surround kind of processing, essentially run a contrast enhancing filter over the image. Now, a number of you were chuckling, so you recognize that there's a disconnect between this fantastically intricate structure and this proposal for what the function is of the circuit. Yeah? In fact, the retina of the limulus, the horseshoe crab that the invertebrate that floats through the shallow waters around Woods Hole, is able to implement this function with just one population of neurons, the photoreceptors. Yeah? So lateral inhibition requires one type of neuron, and one has to then ask, what are the other 49 types for? Yeah? And so our kind of conviction has been for some time that the retina actually serves much more intricate functions. And we've been looking for these functions. We've been trying to identify them and sort them and understand them in order to do justice to this beautiful structure and kind of fill in the gap that is apparent in this paradoxical picture. Well, how would you find out what the retina is really trying to do? One approach that we took was to ask, well, what are the real challenges for the visual system? What are the challenges for the eye and the retina that it maybe has to deal with and has to dedicate some neural circuitry to? And one challenge that is commonly appreciated is the fact that the image on the retina is always in motion. There are three major reasons why it's always in motion. The first is that the subject, meaning you, is constantly moving its eyes around. This is a classic picture from the Yarbus book on eye movements, tracking the movements of the gaze when a subject views this image. And you can see here that the eye executes these very rapid movements that center the gaze on different parts of the image. So these are so-called large saccadic eye movements. Yeah? That's the most obvious way in which the image shifts around on the retina. But it turns out that even when the subject tries to keep his or her eyes still, the eye continues to jitter a little bit, and I'll show you a demonstration of that a little bit later. And these movements are called fixational eye movements. So even when you think you're holding your eye still, the image continues to move on the retina. And anyone who's taken used a photographic camera, you know that it's a bad idea to move the camera while the shutter is open, right? The image gets blurry and you can't see things very well. And yet that's what happens in vision. The shutter's always open unless you blink, but the image is constantly moving on the retina. So we thought that there must be some aspect of retinal processing that deals with this interesting constraint. And finally, perhaps the most obvious source of movement on the retina is that objects will move within the scene. So, for example, this girl might decide to smile one day, and these movements are, of course, perhaps the most important, ecologically speaking, the most important aspect of image motion. When objects move within the scene, allows you to detect predators or prey, or in this case, a love interest. So what I'd like to do today then is tell you 3 stories, essentially, about what we think are retinal specializations for dealing with these movements of the image on the retina. And in each case, I want to give you a demonstration that this actually happens and is an interesting phenomenon. I'll tell you something about how the retina processes this input, and a little bit about the circuits that we've identified that we think are behind it. So let me start actually with the smallest eye movements, these small fixational eye movements. As I said a moment ago, even when you think you're holding your eye still, the image continues to jitter. So why don't you, while I talk here, why don't you fixate on this black spot here for 10 or 15 seconds? And then a little while later, after you've looked at that for some time, you can move your eyes to the white spot, and you should see a little after image of the grating you were just looking at. And if you pay attention, you'll see that that after image moves around a little bit. Yeah? Now, you can move back to the other spot if you didn't see it the first time, so it'll work eventually. The after image, of course, is burned into your retina, so the after image does not move. It's a result of adapting your photoreceptors. But the real image is moving, and in your mind, you think the after image is moving. But it's a demonstration that your eye, even when you're fixating on one point, your eye continues to execute these movements. It also gives you a sense of how large these movements are. They're much bigger than your detection threshold or the velocity of things that you can detect. They're really quite substantial. So one might expect that these movements should seriously affect the performance of your visual system. Let's take a closer look at what this means. So suppose you're a predator approaching this herd of zebras. On the predator's eye, on the retina, the image does this jittering motion, yeah, because the eye is always moving a little bit in the head. By the way, these eye movements result in part from little muscle tremor, in part because the head is moving, in part because you're breathing, and so on. All these components contribute to that image motion. Now, what happens when one of these zebras starts to move, like this one here? On the retina, this is what it looks like. Everything is moving, yeah, but the background is moving with one trajectory, and the foreground, this foreground zebra, is moving with a different trajectory. So for the predator to detect the moving zebra is not a simple task of detecting motion within the image. Any first-year undergraduate computer scientist can write an algorithm for detecting where something moves. Yeah? But this is a somewhat trickier algorithm. You have to find a spot in the image that moves differently from all the other spots in the image. Yeah? So you might think that somewhere in the visual system there ought to be a stage where this differential motion gets computed. A stage that has neurons that are detectors of such differential motion. Neurons that are silent when everything is moving together, but fire when a patch in the image moves differently from the rest of the image. And remarkably, you find these neurons already in the retina. So let me lead you through an experiment. Here I'll show you in detail one experiment to give you a sense of what the actual data look like that we work with. So we project an image onto the retina while we're recording the signals of retinal ganglion cells. Yeah? And we start with an image like this one. So here we have nothing in the background, and then you can think of this as a zebra in the foreground that's jittering back and forth a little bit. Yeah? And we record from a retinal ganglion cell, and we get this kind of response. And what I'm showing here, this is a so-called raster plot. Every tick mark here is one action potential, one spike from such a ganglion cell. And successive rows in this graph are 8 identical repeats of the same jittering stimulus sequence. Yeah? And so here's the average response. So the cell is silent a lot of the time, but it fires these strong bursts of spikes at certain times during this jitter sequence. Yeah? This is just to show that a different animal also has such retinal ganglion cells. Okay, in the next experiment, we jitter the foreground zebra with the same trajectory, but now we also have a herd of zebras in the background, and that jitters with a different trajectory. So this is a situation where the eye is moving, but in addition, the zebra is moving within the scene. Yeah? And so what does this neuron do under those conditions? Here's the response, and it's virtually identical. In fact, it fires at exactly all the same times. I should say this ganglion cell is looking at a patch of the image that's inside here in the region of the foreground zebra. So from these two experiments, one might conclude that the background really makes no difference. Here we have an experiment with no background, and here we have one with a jittering background, and the two results are the same. So we do a final experiment then, I hope you can see this behind the podium, where the foreground zebra again moves with this jittering trajectory, and now the herd of zebras in the background moves with the same trajectory. So this is the ecological situation where the eye is moving, but everything is stationary in the world. And what do you think these neurons will do? They stop firing. Yeah, so the firing is essentially completely suppressed. So these cells are detectors of differential motion. They fire strongly when the object in their receptive field moves differently from the background, but they're completely suppressed if it moves in the same way as the background. Now, ideally, you would like such a differential motion detector to not be specific to zebras. You'd like it to also fire when there's a gazelle or a frog or whatnot. Yeah. This is getting to be a tall order. We'd like this neuron to be selective for the condition of differential motion, but non-selective for what it is that's moving. Well, it turns out these neurons have that property. So here, for example, we changed what the engineers call the phase of the grating. What that means is that we changed all the black bars to white bars and vice versa. So we completely inverted the stimulus and used the same jitter trajectory, and the neurons fire in the same way. The response is invariant to this change in the stimulus. In another experiment, we changed the coarseness of the grating. You can think of this as the adult zebra, the juvenile zebra, and the baby zebra. So a different spatial frequency of the pattern. And again, it doesn't matter. As long as the pattern moves with the same trajectory, you get the same responses. Yeah. So these neurons are both specialized for differential motion and invariant. They don't care about what it is that's moving to a large extent. Let me say also that this is a special population of neurons. So in both the rabbit and the salamander, we found that this involves a population of about 5% of the ganglion cells that have this property. And more recently, we found them in the mouse retina as well. Now, how does this work? Let me give you first a kind of a general picture of how the algorithm works. The center region of the ganglion cell's receptive field is excitatory. When something moves, a grating moves in the center, the ganglion cell receives these brief bursts of excitation, and these bursts of excitation lead to the action potentials that the neuron fires. If the same grating also covers the periphery of the receptive field is inhibitory. So under these conditions, the ganglion cell also gets brief pulses of inhibition. And if the pulses of excitation and inhibition align in time, they cancel each other out and the neuron remains silent. On the other hand, if the surround grating moves with a different trajectory, then these pulses of inhibition will be timed differently. They'll occur at more or less random times relative to the pulses of excitation, and therefore the excitation can break through and make the ganglion cell fire. This is fundamentally how the system works. It's really a time domain computation, if you want. It doesn't involve complicated spatial-temporal computations. Well, now we have to ask, though, what is the circuit that turns the jittering trajectory of an object into this sequence of pulses of excitation, or of inhibition in that case? And here's what we found out about this circuitry. The ganglion cell gets input from many bipolar cells. Bipolar cells, remember, are these vertical interneurons that bring the signals from the photoreceptors to the ganglion cells. Each bipolar cell has a small receptive field, meaning it looks out at a small patch in the world. And the transmission from bipolar cell to ganglion cell is rectified. meaning only if the bipolar cell gets excited does it transmit that to the ganglion cell. If the bipolar cell gets inhibited, then that doesn't work, that doesn't pass through. So you can see now what happens if this object moves. Here's a representation of our black and white zebra. If the object moves, then it will excite some of these bipolar cells. They'll go from black to white, and some other bipolar cell will be inhibited, going from white to black. But only the ones that are excited will transmit that to the ganglion cell. And so now we can understand already why it doesn't matter where the black and white bars are, or whether they are fat or narrow or what. In any case, when the object moves, some bipolar cell will get excited and communicate that to the ganglion cell. Now we've learned a lot of detail about this process. We've been able to record from individual bipolar cells and measure their spatial receptive field and their temporal response properties. We've also been able to map out this rectifier by recording from retinal ganglion cells. And by plugging all these things into a simple model of the circuitry, we can predict now what should happen when the object passes over the retina with a certain trajectory. And here's an example. These are now spike trains recorded from ganglion cells, these brief bursts of spikes that I've showed you already. And these are the predictions from this model for when the spikes should happen. And as you can see, that prediction lines up very nicely with the measured responses. So this model actually accounts not only for the idea that the ganglion cells should fire, but moment to moment for when they will fire exactly. Okay, so that's what happens in the center of the receptive field. How do the ganglion cells get suppressed by motion in the surround? So here what we found is that there's a similar circuit in the surrounding part of the ganglion cell receptive field. So there's a much larger circuitry out here where the same kind of convergence of signals goes onto amacrine cells. Amacrine cells, remember, are these interneurons that form lateral connections to ganglion cells and inhibit the ganglion cells, so they deliver inhibition. And now you can see that if the object and the background move with the exact same trajectory, then this amacrine cell will receive the same excitatory input as this ganglion cell, and consequently its inhibition will arrive precisely timed to cancel the excitation, and the ganglion cell will remain silent. On the other hand, if the object and the background execute different trajectories, then the inhibition will arrive at the wrong time and not cancel. So we'd expect then that under global motion with object and background synchronized, the ganglion cells should remain silent, and in fact that's what is observed. So in recent years, we've learned quite a bit more about this circuitry. We've probed it in many different ways, and we're really quite confident now that this is the mechanism, not only the algorithm, but the mechanism by which the retina computes this differential motion signal. Let me move then to another form of motion on the retina. And this has to do with actual objects moving in the scene, moving in the world. When we interact with moving objects, the speed of processing of the retina matters a great deal. We all know that there's some delay involved in visual processing. In fact, in Tel Aviv traffic, you are much more aware of this even probably than in Boston traffic, that when the brake lights on the car in front of you, you have to very quickly move your foot to the brake. And I'm sure your reaction times are shorter, but ours are about 0.2 seconds or so. And approximately 0.1 second of that is actually in the visual system. It's just the delay of signals flowing from the photoreceptors through the various layers of the retina, through the various layers of your areas of your visual system to someplace, where you actually see that the brake light came on. So approximately 1/10 of a second, roughly speaking, of delay to the higher stages of visual cortex. Now, of course, that determines how much distance you should keep to the car in front of you. But let's think about a different situation here. In 1/10 of a second, this ball flies approximately 5 meters. Yeah, so that's from here to the wall. So if we believe that Roger Federer sees the ball with 1/10 of a second delay, then legitimately he should never hit it, right? Because it should be 5 meters in front of where he sees it. Now, you could argue that learning how to play tennis is all about getting used to swinging the racket in front of where you see the ball. But that's not what it feels like, right? When we catch balls or interact with moving objects like that, we don't have the feeling that we have to swat at the empty air. Yeah? So another possibility, though, is that perhaps our visual system actually presents these objects to us where they are currently, not where they were a tenth of a second ago. Yeah. And so there's been this idea that perhaps part of the function of the visual system is to extrapolate the trajectory of objects so that we can perceive them where they are currently, not where they used to be. There's an interesting experiment that was done that suggested this kind of thing called the flash lag effect, where human subjects are shown a rotating propeller, and whenever the propeller is vertical, these two bars flash briefly. Yeah? So at the time of the flash, this is the physical situation of the display that the human subjects are shown. And then you ask them what they see at the time of the flash, and they always say that they perceive this. The propeller seems to be ahead of the position where it's actually located at the time of the flash. And so this brought up this idea that perhaps the moving propeller is getting extrapolated by the visual system. The visual system effectively predicts where the propeller will be based on the old data from the photoreceptors. Whereas, of course, you can't predict when these bars will flash. Okay. I want to show you a version of this so maybe you can experience this yourself. It's a nice display by Michael Bach. What I'm going to do is I'm going to push the start button, and then this ball will start rotating around the cross, and every now and then the center of the ball will flash yellow. Yeah? And what I would like you to do is to keep your eyes on the cross. Don't follow the ball around. Yeah? Keep your eyes on the cross, and then ask yourself what you see when the ball flashes yellow. In particular, is the ball completely filled with yellow, or do you see something else? Yeah. Ready? So keep your eyes on the blue cross. Do you have a sense of, is the ball full yellow or partially yellow? Which part is yellow, the front part or the back part? The back part, okay. So now you can move your eyes along with the ball, and you'll see that it's always completely filled. Yeah, or I can slow it down a bit. Yeah, this, of course, won't convince you that I showed you the same thing earlier, but anyway. So now if you keep your eye on the cross, you'll probably find that it is completely filled. Yeah. Okay, so this is one of many such illusions, they say, that gives you a sense that there's something strange about the processing of moving objects. And we were interested in finding out what the retina does with these kinds of stimuli. Now, I'm not going to claim that everything you see here is a result of retinal processing, but we'll actually find out that the retina has an interesting role to play. So we essentially did the same experiment on the retina while recording from retinal ganglion cells. We started by presenting the retina with a flashed bar, and we asked what is the response at the output of the retina. And so here's the intensity profile of the bar. It's a dark bar on a white background. So this is space on the retina, position on the retina, this intensity. And now we're looking at the firing of retinal ganglion cells at different times after the flash of the bar. So at first, at 0 milliseconds, nothing happens. It takes a little while. At 60 milliseconds, you start to see ganglion cells firing just underneath the bar. Then the blob of activity becomes bigger, and then it declines again. Yeah? So, nothing special. It's sort of what you'd expect. There's a response delay, and then after that delay, you get a hump of activity that declines again. On the x-axis is different cells, yeah? So, in practice, we recorded from, 50 or so cells, and I'm drawing a line through them so that it's easier to follow, yeah. But yeah, you can think of this as the neural image in the population of ganglion cells. Okay, so next then we use the rotating propeller bar on the retina. And it serves to think briefly what you'd expect from this experiment. So if you put a moving bar on, we know that there are these response delays right there, 60 milliseconds from the input to the retina to the output. So we'd expect that at the output we should also see a moving blob of activity, but it should lag behind the bar at the input to the retina, right? It should be delayed. Yeah? So instead, here's what happened. When we moved the bar, we found that yes, indeed, there is a wave of activity that travels along with the bar, but that wave of activity is a little bit ahead of the bar, not behind. Yeah, it's actually, its center is forward of the center of the bar. And so if you compare the position of the hump of activity from the moving bar to the hump of activity from the flashed bar, the moving bar signal is displaced ahead of the flashed bar signal, even though physically at that moment they are in the same place. So if the rest of the brain is doing nothing but looking at the output of the retina and interpreting it, it would have to conclude that the propeller is ahead of the flash bar at that moment in time. And then if you now reverse the motion of the bar, then of course, with a short delay, the hump of activity moves to the other side of the bar and is again traveling ahead of the hump of activity from the flash. Now, how is this possible? You might ask, how do these neurons, for example, know that the bar is coming? Yeah, how is it possible for the retina to extrapolate this kind of information? So there's no magic involved, of course. These neurons have a receptive field, meaning they are looking out at a certain region of space. And when these neurons start firing, the bar has just entered their receptive field. So that's why we see activity ahead of the bar. But that's not an explanation, because you might ask, why don't these neurons back here fire in the same way? Why aren't they active when the bar is still inside their receptive field? And here, the reason is that there's an automatic gain control that kicks in. When these neurons start firing, the gain of retinal processing gets turned down somehow. And as a result, the response ends much more quickly than expected, purely based on the movement of the bar in the receptive field. Yeah? So this is a temporal effect. The response gets quenched after these neurons become active, and that effectively pushes the profile of the bar out ahead. Now, you can take the words that I just said and convert them into a mathematical model, and we've shown that this simple model that has this gain control feature is actually very effective in predicting correctly the function of the retina under moving objects. I won't go into more detail here, but you might ask now, what is the purpose of this processing? We've already argued that it's useful for tennis, but we didn't evolve playing tennis, so what is the more general utility? And here's an example of an animal that we think benefits greatly from this kind of processing. It's a salamander that just caught a fly. The fly was walking across the rock, and the salamander launched its sticky tongue out and grabbed the fly and swallowed it. And there isn't a whole lot of time to think if you want to perform this effectively. So ideally, the salamander would like to wire the neurons from the retina directly to the tongue muscle so that it can immediately launch out the tongue when the right moment comes. And what we found is that, in fact, we don't know that that's what the salamander brain does, but it could well work that way, because the information that comes from the ganglion cells is immediately useful for targeting the fly. Here's an example of this. I'm going to show you a movie here where we moved a fly over the retina, or actually a small disc, but moving at about the speed of a fly. And in white here is a blob of ganglion cells that fires as the fly moves. In black are the ganglion cells that are suppressed because of this automatic gain control. And as you can see, the blob of cells that fire is really quite nicely centered right on the middle of the fly. Yeah? So there is no effective delay. There's no shift between the response of the neurons in the retina and the current location of the fly. In fact, the simplest thing the salamander might do is just take kind of the center of mass of this white blob of firing neurons and say, that's where I want to launch my tongue. And in fact, that's what we showed here in this calculation. The red dot is the center of mass of this blob of firing retinal ganglion cells, and the blue dot is the current location of the target. And as you can see, when the target moves smoothly, the two are right on top of each other. So the salamander could just launch its tongue at where the ganglion cells say the object is, but when the target makes a turn, the estimate from the center of mass overshoots a little bit, and then it comes back to the actual trajectory. And anyone have an idea where this overshoot comes from? Yeah, it's a consequence of this extrapolation, right? Remember, the retina operates on old data. They are at least 60 milliseconds old in this case. It estimates where the object is going to be 60 milliseconds later. But of course, when the object takes a sharp turn, it can't predict that. And so briefly, the estimate will overshoot and then return to the actual trajectory. And Anthony Leonardo, who did this work, now has his own lab at Geneva Farms, and he's actually taking measurements off of salamanders that shoot their tongue at a screen, and is trying to see whether he can see these errors happen when the target turns. Haim, how much time do I have?
5 minutes.
5? Okay, then. No, I can do 5. I'll just jump over to the next story. Yeah, let me move across this. I want to leave you with kind of a view of the future. Where I think things are going. So we have now in a number of cases, we've identified interesting visual computations in the retina, and we've boiled them down to specific circuits, and we've drawn little circuit diagrams like this one here. I think it's important to actually go through the exercise of drawing a circuit diagram because it, in some sense, embodies everything you think you've learned about the system. It also allows you to make predictions for its function. I would be happy if down the road we could reduce the retina to 15 circuit diagrams, each of which has the complexity of a small radio or something like that. And I think, in fact, circuit neuroscience in general should work towards that goal. We really want to be able to write down the result of our studies in terms of circuits that make active predictions. Now, one difficulty with this goal or this prediction is that unlike the circuits in a radio, the brain doesn't come with little part numbers stamped on the elements. Yeah. In the case of a radio, even if I don't know it's a radio, I can open the box and I can start looking at who's connected to whom, and I'll see a part that has 75152 stamped on it, and I can put that into Google and find out what that chip does, and so on. So there are part numbers and there are descriptions of what the parts do, and my colleague in Japan can work with the same parts and get the same results. So there's this wonderful common language that results from having identified parts. And so far, at least, the brain doesn't come with these part number identifiers. And that's been a real obstacle, not only for any given lab studying the circuits, but also in communicating your results to other labs. You don't necessarily know that you're studying the same neuron when you can only describe it vaguely by what signals it produces. Yeah. So there have been some very interesting developments recently that are about to change the situation, and it looks now as though we're going to have methods that will allow us to put part numbers on neurons, will allow us to identify the cell types in the brain by making them glow in different colors. And here's a picture that went through the press 2 years ago from my colleagues Jeff Lichtman and Josh Sains, in which they modified a mouse so that its neurons glow in 70 or 80 different colors. This was kind of a demonstration that showed that you can make every cell look different. But more important, to me at least, are applications where we can tag specific cell types that we recognize as being cell types, and we can label them individually and then study them in great detail. And I want to give you a flavor of what this can look like. My colleague Josh Sains has recently designed several lines of mice in which different types of retinal ganglion cell are fluorescent, are labeled fluorescently. I'm showing you here 4 examples. In each case, we're looking at a vertical section through the retina. The ganglion cell body is down here. These are the bipolar and amacrine cells. And in between, you can see the dendrites of the ganglion cell going up to a particular level in the inner plexiform layer and ramifying there. This is a different type of ganglion cell that ramifies at these layers. This is yet another type that ramifies at these layers. So these are recognizably different types of retinal ganglion cell. And we now have animals in which all cells of one type are glowing green. Now, of these types, this one here was known before. There's a long literature on this type of ganglion cell, but the other 3 are new. And having these part numbers stamped on the cells allows us to study them very quickly and very effectively. And I'll show you a little bit about what we learned about this type. This was a very nice surprise. Looking in section, it's not that unusual, but if you turn the retina so that you're looking straight down at it, and you look at all the neurons that are labeled in that population, we got this very interesting surprise. So you can see that these neurons they're about 5% of the population, and this is the cell body, and down here is the dendritic tree. And each dendritic tree is incredibly asymmetrical. They all point in one direction like thin pizza slices, and most importantly, they all point in the same direction. Now, just for reference, let me show you what a typical ganglion cell looks like. Here are 3 different types of ganglion cell in the mouse. Typically, the cell body is in the middle, and the dendrites come out, and they go in all directions radially. Yeah. So this is a very unusual shape, and not only are they this asymmetrical, but the whole population points downward on the retina. So with this very asymmetrical shape, we thought, of course, there probably is some asymmetrical function as well. Yeah? And now what kind of asymmetry would the retina be interested in? We're probably a bit monomaniacal about this, but we were thinking about motion again. Maybe they're dealing with image motion or object motion in a particular direction. So we tested the responses of these neurons by passing spots of light moving in different directions over them. And indeed, they are very selective for spots moving in one direction. So here's the result of such an experiment. You move spots of light in different directions, and in one case you get 0 spikes. If you move in the opposite direction, you get a lot of spikes, and in between there's something in between. And this preferred direction was always downward on the retina, and it's the same direction as the dendrites point. So these neurons are specialized to respond to moving stimuli, and specifically to stimuli that move in the direction of the dendrite downward on the retina. Now, why the mouse has this pathway for motion downward on the retina, we really don't know yet. But it's finding this class of ganglion cells actually allowed us to make sense of a piece of work from several decades ago. Because the cells are glowing green, we can also look at where they go in the brain. And in particular, these ganglion cells projected their axons to the superior colliculus and also to the lateral geniculate nucleus. Now, in the superior colliculus, there was a long-standing and somewhat mysterious report from Ursula Dreger and David Hubel that showed that there are many neurons in the superior colliculus that prefer upward motion in the visual field. Here's an example of one such neuron that's very strongly biased for upward motion. And when they plotted the preferred directions of all their neurons, virtually all of them preferred upward motion. And now we know that this computation of upward motion already took place in the retina. These superior colliculus neurons are simply reporting their input, which they receive from this class of retinal ganglion cells. Whereas previously it was thought that this kind of computation happens in the brain, it seems to already be computed in the retina. Again, like Ursula Dreger, we don't really have a clue why the animal has this very strong pathway that is specialized for upward motion. We have some speculations, some are more humorous than others, but maybe we can also talk about them over beer sometime later. To get back to the bigger picture though, having this genetic tag on a particular cell type allowed us very quickly, within 2 or 3 months, to make a lot of progress on understanding their function, the circuits that lead to these ganglion cells, and the places where they project in the brain in ways that I think would have been impossible otherwise. So I used to be a real skeptic about the power of molecular biology and neuroscience, but I've gotten converted, and now I'm, I guess, becoming a preacher, as is the usual course of events. So this is really changing the way we work. This ability to identify cell types is really changing the prospect, I think, for circuit neuroscience. Let me return then to this picture. As I've tried to show you, we've identified a number of interesting visual computations that we can relate to ecologically important problems that the visual system faces. In each case, we find that there are circuits in the retina that are actually seem to be designed for implementing this kind of thing. And we're hoping that over time, this kind of effort will help to flesh out the bottom part of this diagram to really fill it up with functions and circuits that are more commensurate and do justice to the beautiful structure, anatomical and synaptic structure of this circuit. Thank you very much.
Time for questions?
Yeah.
I just wonder, you describe this ability of the ganglion cells by prediction, essentially extrapolating time, as a consequence of some side product of this gain control.
Yeah.
It seems to me that it should be the other way around. I mean, the gain control mechanism should provide you with this prediction ability, which is the main task. So I don't know if it is an explanation or a result, some deeper principle that you want to make good prediction.
Yes, I guess historically the gain control was discovered first, and we would suggest certainly that an important function of it, and maybe the important function of it, is this this ability to track moving objects. And in fact, you find that some ganglion cell types have more of it and others less, and so only certain ganglion cell types are in fact provide this correction for the response delay, and others don't. So I would agree with your viewpoint. I want to emphasize that this all the components in that little model diagram that I illustrated were actually known before, and we showed in some sense that if you put them together and provide the model with an ecologically interesting input, it has this important consequence of extrapolating the motion.
Other cells may attune to different time scales.
Yes, that would be interesting. You mentioned tuning, and that is of course important. I mean, this mechanism is so simple in a way. It involves some spatial integration and some temporal gain control, that it obviously has a spatial scale and a temporal scale to it. So it won't work for all trajectories, it won't work for all speeds. It has a certain preferred domain in which it works well. And Anthony has explored this quite a bit now, the dependence on speed and size of objects and so on. And in these salamanders that target flies in particular, it seems to be attuned in the right, for the right targets that they actually eat. Yeah.
Still, for the salamander, it's okay maybe, but how much of the stability of this picture of Airbus that you see comes from the retina, and how much comes from prediction by the brain rather than by the ganglion cells is the question. Only can you quantify to what extent this property of the ganglion cells could account for stability of the retina?
Yeah. Stability of the image, yeah. I think that Stability of the perceived image, that's something I don't know anything about. So the question is, okay, I demonstrate to you with this grating that your eyes are moving around, and yet you don't perceive the world as moving around, even though it's clear that the world is moving on the retina. So somewhere between the retina and perception, these image motions have to be discarded and canceled out.
Only by the same mechanism of prediction.
Perhaps, yeah. But obviously that doesn't happen in the retina, but it has to happen at some higher stage. I think in a way this problem of prediction is one that probably every neural circuit faces to some extent, right? I mean, neural processing entails a delay. Delays are bad if you're interacting with other agents. And so my guess is that each neural circuit is trying to do something to compensate for its own delays. And that might mean a bit of high-pass filtering, a bit of differentiation or something like that. And what we find then at the level of perception is the kind of accumulated contribution of many of such extrapolation mechanisms.
I'm interested if your team investigated the response of the retina and the rest of the neural system to short light pulses, and more specifically, does the entire vision system operate as an energy integrator, or what's known in electronics as sample and hold, which means retaining the intensity memory of the last pulse until the next one?
Yeah, I think if you make pulses short enough, then this is not my work, but is historical. So if you make the pulses short enough, there comes a point at which the width of the pulse doesn't matter anymore. Only the integrated intensity matters, the total number of photons. Yeah. And short enough means essentially short compared to the impulse response of the receptor neurons. Yeah. So these photoreceptors, the first neuron that is actually absorbing the light, they have an impulse response in mammals of about 30 millisecond width or so. Yeah. So for, excuse me?
That's our experience.
Yeah. And so for pulses shorter than that, the visual system can't detect the difference. Yeah. Pulses longer than that will start to look like steps of light rather than short pulses. Yeah.
About the processing that you mentioned that goes on in the eye, how much of it is hard written and how much can be trained? I heard a lecture about a year and a half ago of a study done on computer gamers and how their visual response is so much quicker than normal people's vision response. So whether that is tied into the processing going on in the retina or in the brain.
My guess would be in the brain. The retina is, compared to other stages of the brain, the retina is considerably hardwired. There are some processes in early development, in the first few weeks of life, where you can affect the wiring of the retina by, let's say, raising an animal in darkness or something like that. But that's actually, the effects there are minor, much minor compared to the effects that you get in the visual cortex from a similar perturbation. So my guess is that the effects of practice are not in the retina. There are, on a more basic level, the retina does adapt its function to the visual environment to a great extent. So we all know about the light and dark adaptation. When you move from a dark room into the bright light and vice versa, it takes a little while and then you can see again. But there are more subtle forms of adaptation having to do with watching moving displays. If you go from one type of motion to another kind of motion, in the beginning you are unable to differentiate or detect as well as after some period of adaptation. So there are aspects of retinal function that adjust to the visual stimulus on the time scale of 1/10 of a second to several seconds, but I wouldn't, they're not the kinds of practice effects that you were talking about.
Related to this, so